I'm sure most of us have seen or played Galaga. It's the old arcade game where you take your spaceship and you move it around side to side and shoot lasers at things to blow them up. Well, what if you could take that same idea of shooting lasers at things to blow them up and use it to cure Alzheimer's? Well, we can't, but a recent headline suggests this might be a possibility in the upcoming future. But to understand this research, we first need to understand a little bit about the structure and function of proteins. Proteins are large molecules consisting of amino acid chains that serve a variety of functions, from assisting with DNA replication to providing the structural support to cells. They're grouped into four main types. The first type is the primary structure, which is just the chain of amino acids. The second type is the secondary structure, which is the coiling of amino acids to create these alpha and beta sheets. The tertiary structure is the folding of alpha and beta sheets to create a 3D structure. And the quaternary structure is the ultimate structure of the macromolecule that's formed through interactions of multiple polypeptide chains. Amyloids are aggregates of proteins that fold into a specific shape, allowing many beta sheets, pictured here, to overlap, which creates these tightly packed fibrils that are insoluble and resistant to degradation. In Alzheimer's disease, these accumulate into plaques in extracellular spaces and in blood vessels, ultimately leading to the disruption of nearby cells. The recent headline that reported the potential use of lasers to treat Alzheimer's references a study conducted by material scientists at Tokyo's University of Science. In this experiment, researchers used free electron laser technology to direct a high-energy laser beam at a thin peptide film, which produced holes in the peptide film and significantly lowered the proportion of beta sheets, which, as I mentioned earlier, overlap to create the structures involved in Alzheimer's disease. What's interesting, though, is that Alzheimer's was only mentioned once in this entire article. This research was actually proposed to be used in the manipulation of biosynthetic materials, which are basically just biosynthetic structures. A different group of researchers focused on various biosynthetic structures that can spontaneously form, and these are just a few of the structures they came up with. Some of these structures, though, are so strong that something as powerful as free electron laser technology is needed to break them down. Researchers are hoping to use these structures for things like tissue engineering, slow-release drug delivery, and repairing spinal cord injuries. But the idea of treating Alzheimer's with lasers builds off a study that was conducted in 2013, in which researchers found that beta amyloid proteins have a specific multi-photon absorption that is different than other proteins, meaning the researchers found a way to distinguish between various types of protein based on how they absorbed light. And this ability to distinguish various types of proteins would allow us to selectively disrupt the dysfunctional beta amyloid proteins without impacting other healthy proteins. Okay, so we can visualize the type of protein with multi-photon techniques, and we can eliminate those proteins with free electron laser technology. Perfect! Problem solved! No more Alzheimer's disease! Well, you might be wondering how exactly we would shoot lasers into the brain of live patients. And that's a valid question that we don't have the answers to. However, a group of researchers was able to map out a vascular region in a rat brain and use laser technology to selectively rupture a deep blood vessel within a live rat without impacting nearby tissues or blood vessels. And although these aren't beta amyloid proteins, microvessel dysfunction also contributes to the cognitive impairment that we see in Alzheimer's disease. So this was extraordinary in demonstrating the accuracy of lasers, but this procedure wasn't a walk in the park for rats. It was a full-on brain surgery, with the part of the skull above the brain area of interest being removed, the dura mater covering the brain disrupted, and a metal frame pictured here glued to the brain itself. And you can see how complicated the laser system is. So, at this point, we've got an invasive procedure for shooting lasers into the brain that would allow us to selectively disrupt structures involved in Alzheimer's disease. So brain cells normally fire at a rate between 25 and 80 times per second. This pattern is known as gamma oscillation, and it is thought to be essential for critical thinking processes and is known to be dysregulated in Alzheimer's patients. So, a few years ago, researchers decided to use optogenetics to activate neurons in the hippocampus, a brain region involved in Alzheimer's, which makes sense because of the characteristic forgetfulness we see in Alzheimer's patients. But they wanted to activate these neurons at a specific rate of 40 times per second. to artificially restore the normal gamma oscillation firing patterns. They found that when they did this, they were able to significantly reduce levels of beta amyloid protein. The EYFP here stands for yellow fluorescent protein, and this was just a control group that did not receive the channel rhodopsin virus that activates neurons. As you can see, there was significantly less that fluoresced in the experimental group that received the 40 Hz light stimulation. They also observed a simultaneous increase in microglia, which are a type of support cell that play a large role in regulating and maintaining neurons, and by staining microglia and beta amyloid together, the researchers were able to visualize the microglia engulfing the beta amyloid protein. So by activating neurons in the hippocampus, the researchers were able to get the brain to attack these dysfunctional beta amyloid proteins naturally. That is pretty cool. But optogenetics also involves brain surgery because they had to inject a virus into a specific region of the brain and implant a fiber optic probe into that region of the brain to emit light. Maybe it's less invasive than getting a metal frame glued to your brain, but this is still brain surgery, and this is not good enough for humans. So the same researchers also knew from previous literature that visual light stimulation could drive gamma oscillations in the visual cortex. Since the visual cortex is the region of the brain that responds to light, when a light flashes 40 times per second, the visual cortex responds with activation 40 times per second. So knowing this, the researchers placed mice in a dark box and flickered a light on and off 40 times per second. And when they did this, they again saw that they were able to significantly reduce levels of beta amyloid protein by about 50% in the group that received the 40 Hz light stimulation. And in this case, there was no brain surgery necessary. They just put mice in a dark box and flickered a light on and off a few times. Additionally, they observed the same microglial engulfing patterns in this treatment. Beyond this, as if that wasn't enough, the researchers also observed a substantial decrease in the amount of tau protein, which is the second of the two major pathological hallmarks of Alzheimer's disease, the first being the buildup of beta amyloid. Like beta amyloid, tau proteins also accumulate in the brains of Alzheimer's patients, but they essentially form tangles that strangle the neurons to death. Tau pathology is also seen in CTE, or chronic traumatic encephalopathy, which is the neurodegenerative disease that many boxers and football players suffer from because of repeated blows to the head. There are limitations to this research, though. The first limitation is that this decrease in beta amyloid and tau protein only occurred in the visual cortex, since that was the region of the brain that responds to visual light. Here's a graph that shows the amount of beta amyloid in the hippocampus, the region of the brain that we would like to reduce these beta amyloid proteins. And as you can see, there was no change in levels of beta amyloid before and after treatment. This is obviously a substantial barrier to overcome, since this treatment isn't influencing the regions of the brain that are implicated in Alzheimer's disease. Secondly, this treatment didn't even last a full day. Here's a graph that shows the amount of beta amyloid protein in the visual cortex, where we saw the decrease in beta amyloid proteins, and you can see that over time, the levels of beta amyloid returned to baseline just 24 hours after treatment. So this isn't quite the miracle cure, at least at the moment. But in the meantime, researchers are working tirelessly on developing new drug therapies that can selectively target beta amyloid and tau proteins and other techniques that can potentially be beneficial in treating Alzheimer's disease. But for now, lasers should continue to be used to blow up spaceships and manipulate biosynthetic structures, not the brains of live Alzheimer's patients. Thank you.
It's Galactica.
It's Galaga.
Is
That's
it Galactica?
Battlestar Galactica. Galaga. You're gonna crank this out.
I'm excited. Oh, great, that's such a plan.
You got somewhere to be?
Yeah, I have to be at a meeting.
When?
Like now-ish.
Where?
Here.
That's
When?
okay, you don't have to say.
Alexis!
Nadalski?
I'm sure most of us have seen or played Galaga.
I'm sorry.